This is my Chapter 7 presentation. So first I'm going to talk about the properties of virus. So viruses are everywhere and they're very small and they can infect literally any type of thing from bacteria, fungi, plants, animals, even algae. And they're extremely small and very abundant. So like in it, just a small drop of lake water, there could be up to like 250 million, I think is the example. There's huge amounts of viruses everywhere, and they're very important for how the earth has evolved and shaped the life that's on it. So viruses, they are not cells, but they don't have organelles, they don't have cytoplasm or any of the other stuff that most other things need to function. So they're like little intracellular parasites, and it's a fancy way of saying they basically just live inside another cell and reproduce. And their structures are very simple, which is like a capsid, and this is like the shell of the protein, and it's wrapped around the nucleic acid core. And cells, unlike cells, viruses, they only carry RNA or DNA. So cells, they usually have both, but viruses only have DNA or RNA. And they can either be double-stranded or single-stranded, and it differs how they handle their genomes. So viruses, they can either be alive or dead, which is actually just like inactive or active. So there's no really way of classifying if they're alive or dead. So we just say active or inactive. So when a virus is outside of its host, it's basically just there. It's like turned off. So there's no activity, no reproduction, metabolism, there's nothing going on. But once it gets into a host, the switch basically flips on and the virus starts doing what it's supposed to do. So viruses, they cannot reproduce on their own. They have to have a host. So because they don't have any of the enzymes or organelles to do metabolism or reproduction or stuff like that, like other living cells. So they get into a host and basically like take over the cell and turn it into what they need to live. So viruses also show host specificity. And this means they are like particular on what they inject. So their surface molecules are like they only go on to specific cells, and that's why some viruses, they don't infect humans, but they infect animals, and also why viruses only target some parts of our organs or tissues. And so I'm just going to circle back. Are viruses alive? Most people say no because they can't live on their own, but they also are still functioning. Once they get to a host, they're still able to reproduce and all that stuff. So that's what we call active and inactive. And now I'm going to talk about viruses and other microorganisms. So here's a picture showing all the different sizes of bacteria. So they are very small, and they can only be seen with a light microscope. So an electron microscope is needed we can't see them with the light microscope, an electron microscope. That is what we need to see viruses, and you can view their shapes and structures. But viruses are very small, much smaller than bacteria. You can fit 2,000 bacterial viruses, could fit in just one bacterium. And their size can vary. So as you can see, the hemoglobin molecule is very small compared to E. coli, which is huge bacterial cell. And the smallest known one is a paravirus, and it was measured up to about 0.2 micrometers. Now I'm going to talk about the components of a virus. So viruses we already discussed, they're not really like cells at all, and they don't have ribosome, cytoplasm, organelles. They can't make proteins on their own. So instead, their basic structure is just genetic material. So it's just genetic material, and it's wrapped in the protein coat. And together, the capsid, protein coat, and the genetic core make a nucleocapsid. So the nucleocapsid, just the capsid and the nucleic acid core. And some viruses also have envelopes, and this is where they'll like steal off some of their host membrane, and this creates an envelope around themselves, and this helps change how they interact with and exit host cells. So now I'm going to talk about the naked viruses versus enveloped. So these are like the two styles that you see viruses in. So naked viruses just have the nucleocapsid, and it makes them tough and stable into the environment. But envelope viruses, these have the extra envelope membrane covering and that they've took from the host, and it actually makes them more flexible and able to shape into different shapes, not rigid, very flexible, and they're also more delicate. So if they like get outside of their host, they're easily destroyed or can even dry out. And it also changes the way they infect cells. So naked viruses, they just inject their genetic material, but envelope viruses, they usually fuse with their host to get inside. So I'm going to talk about spikes. Both naked and envelope viruses have these surface proteins called spikes. So they're like little rods that stick out, and they allow the virus to stick onto their specific host. And without these, they wouldn't be able to attach and get inside and couldn't affect their host. So that's why these are important, and they also determine which species or tissues that their virus wants to target. So an example is the flu, influenza. It uses their spikes bound to the respiratory tract. So now I'm going to talk about the structure of capsid. So the capsid, we've already talked about, it's the protective protein shell, and it's made of repeating protein units. These are called capsomeres. And the capsomeres, they just naturally self-assemble into shapes. So there is a helical, which is like spiral, and that wraps around the genetic material. And then there is naked. So naked versions these are more stiff and rigid. And then we also have the icosahedral, and this is like a more complex one. It's 20-sided with 12 different corners, and it can be naked or enveloped. And good examples are adenovirus and herpes. So there's also complex capsids. These are weird, and they're mostly found in the bacteriophages, and they look like little balls with stuff sticking out of them, like very different from the other two. And now I'm going to talk about the viral envelope. So we talked about this a little bit before, but when viruses with envelopes, they exit a host, they don't just go away. So they'll bud off from the cell and take some of its membrane, and this is what they use for their envelope. So they can use the cell membrane, nuclear envelope, or the ER to insert their own proteins into that membrane and create these unique viral spikes. So the result is an envelope that kind of looks different from the other two, and because their membranes are flexible, the envelope viruses, they come in many shapes, like long, round, different, thin, yes. So now I'm going to talk about viral genomes. So viral genomes, they're different from cellular genomes. So cells always have DNA and RNA, but viruses can only carry one type. And also their genomes are way smaller. So some have as few as 4 genes, but like Pandora virus can have over 2,500. And for DNA viruses, these can be either single-stranded or double-stranded or arranged linearly or in circles. So that's what they look like. But RNA viruses, they can be double-stranded or single-stranded. And single-stranded RNA comes in 2 different ways. Positive sense, which is ready to translate. And an example of this is coronavirus. But then there's other ones like the flu, and they split their genome up into pieces, and that's called segmented RNA. So now I'm going to talk about retroviruses. These are special because they use an enzyme reverse transcriptase, and this turns RNA into DNA and then inserts it into the host genome. And HIV does this. So now I'm going to talk about other viral components. Some viruses, they have prepackaged tools, and though they don't have full metabolism, they can bring along enzymes, so like polymerases for making DNA or RNA, and replicases for copying RNA, and then there's also reverse transcriptase in HIV, and this flips RNA into DNA. So now I'm going to talk about the animal virus cycle. This has 6 basic steps. So there's adsorption, and this is when the virus attaches to the cell, it finds the right one that it wants to and latches on, and they use specific receptors that's on the surface to tell when to attach and what to, and it's all about being specific. So this is like HIV. HIV, it targets helper T cells, and without the right receptor, the virus just wouldn't infect it. So then there's penetration, and this is when it enters the cell. So once the virus is attached, it wants to get inside, and some go through endocytosis where they basically get swallowed into it, but others will fuse their way into the envelope and attach directly with the cell membrane, and they just basically want to get inside the target. So then there is uncoating. So this is after they have entered the cell, the capsid or envelope, it'll break down and it'll free the viral genome into the host cell. So it's like unpacking, uncoating the capsid or envelope. And it basically wants to get into the cell and hijack all things so that it can take over. And without uncoating, the genome would just basically be useless and it would just be trapped there. So next is synthesis. This is when the host makes its viral proteins and genomes and takes over. So sometimes it will redirect the host's transcription translation, and it'll carry out viral proteins instead. And it can also make copies of the viral genome. Instead of doing its normal job, the host cell is basically turned into a way that the virus makes their proteins, their genome, and takes over. So now there's assembly, and this is basically viron construction. So all the parts that's being made will come together and get packaged up and be carried out into new functional viruses, which are virons. And then lastly is release, and this is how the virus leaves. So some viruses, they just use lysis, which is when the cell bursts, but then others do a slower method, so they'll use budding, and this is like more gentle. It steals a bit of the membrane into their envelope, and budding lets the cell stay alive longer and keep producing viruses. So now I'm going to talk about, actually I'm going to skip this because I don't have time. I want to get to the other things. Okay. So now I'm going to talk about the cytopathic effects, or CPEs. So these are visible changes that you can see under the microscope. So you can see inclusion bodies or syncytia. And inclusion bodies, these are just like clumps of virus particles. That's what's shown in red in the picture with the arrows. And it can come from damaged organelles inside the cell or virus particles, as I just said. And syncytia, these are multiple cells that have like fused together into a bigger like cell with many nucleuses. So CPEs are important because they help identify which virus is present. So now I'm going to talk about persistent infections. So not every infection kills the cell right away, but there are different ways that these infections happen. There's latency, and this is when the virus hides inside the cell and it's not very active or produce new viruses. So the genome's still there, but it's not causing any effects. So then there's chronic infection. Oh, an example of latency is like when you get a cold sore after a long time. So the herpes can stay inside you and not show up, but then show up 5 months later. So then there's chronic infection, and this is instead of hiding, the virus keeps making new particles at a slow, unrecognizable level. So it's like measles can exist in your brain cells and eventually later on be fatal. So then there's provirus, and this is when the viral gets integrated into the host's own DNA, and since it's part of its genome now, every time the cell divides, the virus goes along with it. So now I'm going to talk about oncoviruses, and oncoviruses, they are a way of, it's like a type of permanent infection, so they transform the host cells in a permanent way, and they're linked to oncogenesis, and this is like the process of cancer development. So there's two strategies to do this. Is oncogenes, some viruses, they carry cancer-causing genes in their own genome, and when they infect a cell, these get expressed